Today I'm going to talk to you a little bit about species' geographic response to past climate changes. In my dissertation, I was focused on looking at species' response to climate change, both geographic response and morphologic response. I've also looked at community responses to climate change as well as environmental gradients. But today I'm just going to present to you one of the chapters of my dissertation and try to show you how I got to be interested in working on the evolution of multivariate systems, which I will present to you some preliminary ideas I have about moving this work forward. To get you start thinking about how dynamic our climate is, I put two maps here. This is the mean annual temperature in our modern climate, and this map shows the mean annual temperature during the last glacial maximum. So you can see that we've had drastic changes in climate over just 18,000 or 20,000 years. So the main question that drives my research is, how do species respond to climate change? Well, this is particularly relevant because of the impending climate change that we are going to experience over the next century. Here's a graph showing the change in mean annual temperature on the y-axis and then year on the x-axis. And you can see this gray curve, this black curve here with gray confidence, shows the change in mean annual temperature over the last 1,000 years. And these colored lines show the predicted change in mean annual temperature over the next century. Now, there's some confidence here. So the change in mean annual temperature over the next century is projected to be between 1.5, and most of the models project about 4 degrees increase. But some more drastic estimates are 4 degrees increase to 6 degrees increase. Looking at a longer time scale, we can see that the change in mean annual temperature, again here on the y-axis, and year on the x-axis. Here, year is presented on a log scale. So this side is millions of years before present, and this side represents thousands of years before present. You can really see that the general trend here is one of global cooling throughout the last approximately 65 million years, so through most of the Cenozoic. It's punctuated with these shorter events of global warming. If I project onto this graph estimates of the change in mean annual temperature over the next century, we can see that in Earth's history, we haven't had a climate that has been that hot since the Miocene, about 15 million years ago. And if we project the more drastic estimates, we have to return to a climatic state similar to one in the Eocene. So given this dramatic fluctuation in climate, I'm really interested in finding out how species might have responded to this fluctuation in climate. One approach that I take to looking at how species respond to climate change is by using species distribution models. And I'm going to try to show you how I projected these models through time, discuss various modeling components, as well as show you 2 examples using North American squamate reptiles. The first example, I'm going to use a genus of rattlesnake, Crotalus, looking at glacial interglacial fluctuations on a short time scale, so over the last 320,000 years. Then I'm going to extend back into time the past 20 million years and look at a little bit longer time scale and see how a genus of spiny lizard, this is Scoloporus, respond to the overall trend of global cooling. Then I'm going to present some really preliminary ideas on how to move this modeling forward into really improve the modeling of phylogenetic changes in species distribution models. So a species distribution model takes species occurrences as well as environmental maps or environmental layers, and combines this information to try to locate the suitable habitat of a species in geographic space. This is a map showing species occurrences or individual occurrences within a species. And we can extract from this map, if we overlay on this map environmental layers, we can extract all the environmental variables that we're interested in at each geographic point. And we can project those points into a climatic space here. So each one of these red points correspond to each one of these red points. So this is geographic space. This is climatic space. And I've shown here 3 variables: annual precipitation, annual mean temperature, and mean diurnal range. So this is a 3-dimensional climate space. And in order to understand where the suitable habitat of a species might be located, or to estimate the suitable habitat, we apply some algorithm to these species occurrences. And what I'm going to talk about today is a climate envelope. So this algorithm takes the minimum and maximum, or the 5th and 95th percentile of each variable within this multivariate space, and puts a cube or a cuboid, an n-dimensional space, around the occurrences. So any space here, the climate envelope is represented by this green cube, and any space within this green cube would be considered by this model estimations of suitable habitat of the species. So this algorithm is called the BioClim algorithm. There are many other algorithms to look at or to use in species distribution modeling, but I'm not going to discuss those today. There are many variables that we can use that are available to model the suitable habitat of a species. I use a suite of variables called bioclimatic variables. They were described by Nix in 1986. These bioclimate variables are describing means and extremes of temperature and precipitation. And some examples, a few examples, are mean annual temperature, maximum temperature of the warmest month. So this is an example of an extreme. Temperature seasonality is an example of some variance of the temperature. Annual precipitation and mean precipitation of the driest quarter. So these estimates are at monthly, quarterly, and annual scales as well. There are 11 other variables that were described. And so this is a suite of variables that are supposed to be biologically meaningful for species. There are limitations to this method that I'd like to acknowledge. So whenever we talk about these models, we really want to understand what they're representing. So these models do not incorporate biotic interactions. Those are interactions between species or intrinsic component variables or characteristics of a species, such as dispersal ability. There's a working group here at NIMBioS that just started a few weeks ago that is trying to incorporate these biotic interactions into species distribution models. So we might be able to include those in the future. There are also geographic barriers to dispersal, such as unsuitable habitat in between fragmented habitats that might be suitable for a species, and these models don't consider those fragmentations. And there's also non-analogous climate. So these models are calibrated on modern climate, and there are suites of climate combinations that are not represented in the modern climate. And so these models typically don't do well in non-analogous scenarios, although the BioClim model is the best to approach this non-analogous problem. So what we're modeling is not actually the species distribution. So it's called the species distribution model. But what we're modeling is equivalent to the climatic niche of a species, the environmental niche of a species, or the suitable habitat of a species. So trying to locate the suitable habitat of a species through time, there are 2 basic approaches that have been used, and those are projections and reconstructions. So if we look at the first approach, it's taking a climate envelope model that is calibrated in modern climate And we can simply project that model onto a map that represents a paleoclimate. And we can locate the suitable habitat of a species during another time interval. And this is an example of this suitable habitat being projected onto a map of the last glacial maximum. So you can see that this range is a bit restricted from the previous example. But this method does not allow any sort of evolutionary change in the climatic envelope. So the second approach is an ancestor climate envelope reconstruction. And it takes phylogenetic information, or the relatedness between species, and it uses that information along with extant data to estimate ancestor nodes, or hypothetical ancestors of these extant taxa. This is a phylogeny, and the y-axis typically represents time. And here, this is a figure just showing that there is a species represented by a red point, one represented by a blue point. They each have their respective climate envelopes, and we can estimate the ancestor climate envelope given extant information as well as this phylogenetic relationship. So that's resulting in this purple climate envelope. Now, just to reiterate, this top approach assumes a completely conserved climate envelope, and this bottom approach, there's no projection of the envelope onto a concurrent paleoclimate map. So this approach typically occurs in climatic space, and there's no mapping that onto into geographic space. So this part of my dissertation, I really wanted to tie these two methods together, not only to obtain paleoclimate maps or paleoclimate estimations at any given time interval, so we could map these ancestor nodes onto them, but also to find any potential ancestor climate reconstruction at any horizontal slice along this phylogeny. So not only at nodes, but along the lineage. So the model that I built, I called it a paleophylogeographic species distribution model, a PPGM. It uses phylogeographies in order to look for the species distribution model in the past. And this model has two main components. One is an estimation of paleoclimate for any given time interval, and the other is an estimation of the ancestor climate envelope. In order to estimate paleoclimate, I start by using a general circulation model, which is a computationally intensive numerical model of atmosphere and ocean circulation. They use geological evidence to validate these models, such as lithology, stable oxygen isotopes, as well as fossil occurrences. This map is an example of an output of data compiled from the output of one of these models, showing the mean annual temperature at the last glacial maximum. So we have pretty robust estimates of what the climate was like for these general circulation models. So again, this is the estimate of the mean annual temperature at the last glacial maximum. And we also know what the climate is like today. So we have estimates, or we know, we have observations of the modern climate. But to understand or to estimate paleoclimate at time intervals in between these general circulation models and modern climate, I've used a stable oxygen isotope ratio curve to interpolate between these two end-member climates. So stable oxygen isotope ratios are highly correlated with the change in mean annual temperature. This curve here, on the red parts of the curve, show interglacial climates, and the blue parts of the curve show glacial climates. This curve goes back the last 150,000 years. And this point on the curve is the last glacial maximum, and this first point on the curve represents the modern. So you can see that these two estimates of the stable oxygen isotope really flank these, the glacial interglacial fluctuations. Okay, so to estimate ancestor climate envelopes, I've used phylogenetic comparative methods. Specifically, the parameters that I've estimated are different from traditional parameters. So traditionally, you would take the distribution of trait values of a species and reconstruct the mean of that value at an ancestor node with some confidence around that node. If we make this traditional implementation, we only have one value with some confidence of what that climate characteristic would have been for the ancestor. So in order to get some range of climate that that ancestor would have occurred in, Graham et al. In 2004 and Hardy and Linder developed a method called maximum encoding. And this method, instead of using the parameter mean, uses a parameter minimum of a distribution separately and independently from the maximum of the distribution. So at each ancestor node, we have a range of possible values in which that ancestor could have occurred in. Now, in order to estimate that node, I've used a generalized least square regression by Martins and Hansen in 1997. This regression estimates the grand mean, which is the most recent common ancestor here, with a generalized least square approach. So this grand mean incorporates the shared similarity. This var y is the shared similarity between tips, the phylogenetic similarity. And it's a weighted mean. So it's a weighted mean of the tip traits. And those are the extant values of the species. We then calculate a matrix W that is a combination of the shared similarity between the tips and as well as the shared similarity between each tip and node. And then we get what we want to estimate, really, which is A, which is the vector of ancestor node traits. So A represents these internal nodes here. And in this final equation, you'll notice epsilon here. This is the error structure. And in the error structure, we can incorporate some model of evolution. And in the first example that I'll show you, I've incorporated a constant rate Brownian motion model of evolution. But that we can incorporate other models of evolution into this error structure. OK, putting this all together, we have estimates of the suitable habitat of a species in modern climate, as well as their phylogenetic relationships. So we can estimate their ancestor climate envelopes. We also have paleoclimate estimates at any given point that we have a stable oxygen isotope estimate for. And we can project these onto those paleoclimate maps. But we're not only restricted to these tips and these nodes. We can use an interpolation to find out what is the most likely estimate of an ancestor envelope as well as a paleoclimate at any given slice or horizontal slice along this phylogeny. OK, so for my first example, I'm going to turn to rattlesnakes. I am looking at the fluctuation in suitable habitat of a genus of rattlesnakes over the last 3 glacial interglacial cycles. This is equivalent to the last 320,000 years. It is a North American genus, except for one species that occurs in South America. And today, it occurs north of the southern extent of the last glacial maximum ice sheet. So we know that these snakes don't live on ice. So they've definitely fluctuated their distributions since the last 20,000 years or so. And also, within this genus, there are several species that are near threatened, vulnerable, or endangered. So it'd be important to look at how their past fluctuations in suitable habitat compare with future projections. So here is a map showing the modern projection of suitable habitat for 3 rattlesnakes. I'm going to use these 3 rattlesnakes as an example through the next few slides. So here's the phylogenetic relationship between these 3 rattlesnakes. The green is the timber rattlesnake, the red is the western rattlesnake, and the blue is the Mojave rattlesnake. You can see that these species diversified millions of years ago, and this simulation is over the last 320,000 years. So we're just barely looking at the tips of this phylogeny here. This black curve here is the southern extent of the last glacial maximum. It's a fixed visual reference. So this map is an animation. It's going to scroll back in time at 4,000-year intervals over the last 320,000 years. But this line will remain constant. And this curve over here shows the stable oxygen isotope ratio in which the animation is showing. So follow this yellow point down the curve, and you'll know where we are in the simulation. So now I'm going to hit play. And you can notice that during glacial climates, these distributions really contract and fragment in sort of complicated ways. And whenever we reach back to the last interglacial climate, we can see that these distributions start to coalesce and expand and also move north. So we'll see the same pattern through the next 2 glacial interglacial cycles. So we'll go ahead and move on. Quantifying these geographic shifts in suitable habitat through time, I've calculated the change in the geographic center of the distribution. This is the average change for all the species in the simulation. And this calculation, the geographic center, can be replaced, of course, for areal extent and also northern extent of the distribution. So whatever characteristic of the distribution you're interested in can be quantified, of course, because we have these estimates in geographic space. This is on a log scale. So this is on the y-axis is the log change in geographic center. And on the x-axis, we have time. So we're going from present on this side of the axis back 320,000 years. Each one of these points represents one of the animations that you saw. So these quantifications consider both the ancestor reconstructions and the changing climate. We can hold one of these 2 components of the model constant and run the other one, which is what I've done. So first, holding the ancestor reconstructions constant, which is equivalent to just modeling the modern suitable habitat of these rattlesnakes over the last 320,000 years of changing climate. We get the change in geographic center very close, following very precisely the change of the full model. Now holding climate constant and running this model with just the change in the ancestor state reconstructions, we can see that the geographic distributions fluctuate very little given the change in ancestor reconstructions. We can calculate the average rate of displacement using the full model. And so using all pairwise comparisons of these time points, I've calculated the average rate of displacement is 0.0023 kilometers per year. And we can compare this rate with future projections of the geographic displacement of these distributions. So projecting the modern suitable habitat, which is here in this map, onto temperature projections from the IPCC in 2007 of a 1 degree increase in temperature results in a 0.43 kilometer per year rate of geographic displacement. And projecting it onto a climate that's 6 degrees Celsius warmer than today, we get very contracted distributions, and we get 2.42 kilometers per year average rate of displacement. So the rate of displacement over the last 3 glacial interglacial cycles is 2 to 3 orders of magnitude smaller than it will be over the next century, or modeled to be over the next century. Okay, looking back at the time scale and the change in mean annual temperature, we've been focusing on these glacial interglacial fluctuations here. So really cyclic climate. And I wanted to investigate how species might respond to these climatic changes over the last 20 million years. So I turned to another genus, and I've looked at 1 million year increments of this changing climate to get sort of a macro perspective. So in this example, I've used spiny lizards, looking at North American genus that has more than 100 species described within this genus. They've already exhibited cold climate, high elevation adaptations, such as the evolution of viviparity, many times within this genus independently. And there's also been recent phylogenetic work by Adam Lachey and John Wiens, which provides us with a good idea about the phylogenetic relationships within this genus. Here's a stylized phylogeny from Adam, built from Adam Lachey's phylogenetic hypothesis in 2010. It contains 53 species. These tips are species groups, so each one of these tips can contain many species within that name on the tip. The black bars on this phylogeny represent 95% credible intervals of divergence. So this phylogeny is dated, and it has these estimates of variance within the divergence time. And the most recent common ancestor occurred around 20 million years ago with some estimate of divergence. So what I've done is I've projected all of these lineages through time at 1 million year intervals over the last 20 million years onto paleoclimate estimates. And instead of showing you individual species fluctuating through time, I've added them all up, and I'm showing you the extant lineage richness through time. So at each geographic location, the heat of the color, so the more red the color, the more number of lineages occur at that site. The gray points on this map are fossil occurrences, and these are fossil occurrences that are dated within the 1 million year time bends. So these are all the fossils that occurred within the last 1 million years. So this is also an animation. It's going to scroll back in time, and you can watch how the estimates of the suitable habitat sometimes line up with these fossil occurrences popping up. And other times in the animation, you can see that the fossil occurrences do not align with these estimates of suitable habitat. See another one popping up here. Okay, to try to investigate why these models are not lining up with the fossils, I've plotted in trait space through time what are the ancestor reconstructions. And this is one example using mean annual temperature. So the tips of this graph here at 0 million years ago represent all observations or observed occurrences of these species. And everything behind it is an estimated ancestor reconstruction based on the tips as well as the phylogenetic relationships. Here on the y-axis, we have the mean annual temperature. And the red reconstruction is reconstructing the maximum of the mean annual temperature. And the gray reconstruction is the minimum of the mean annual temperature. So you can consider any place in between these two estimates to be the estimate of suitable habitat of that species, or the climate envelope of that species. So I'm going to project onto this graph the fossil occurrences. And we can see that the ancestor reconstructions do not overlap with fossil occurrences. So more recent fossil occurrences are well within the genus estimates of suitable habitat. But these older fossil occurrences are not within the estimates of suitable habitat. So one way that I implemented to try to look at the better reconstructions of suitable habitat in the past is to substitute the grand mean of these reconstructions in the generalized least square regression for the fossil occurrences that occur 20 million years ago. So I'm going to fade out these reconstructions, and on top in bold, you're going to see the new reconstructions with the heuristic implementation. So you can see that here, this this last node, most recent common ancestor, has been forced to the fossil occurrences. And we can look and see what the ancestor reconstructions really look like. They really look different, more directional, if you force these reconstructions. But this isn't a very satisfying way to reconstruct these ancestor envelopes. So what I'm going to talk about now is some possible improvements to modeling these phylogenetic changes, especially in this multivariate context of a climatic niche. So let's return to the slide on phylogenetic comparative methods and briefly remind you that the parameters that are being estimated are the minimum and the maximum of each distribution. And we've only implemented here a constant rate Brownian motion model. So there are really two things that we can do to improve these estimates of ancestor climate envelopes. And that's consider a better evolutionary model. For example, the model of a constant rate Brownian motion does not allow for selection. And as you might have noticed, that in the change in mean annual temperature graphs, the temperature over the last 20 million years is cooling. So we have this really large global trend of cooling, which would imply selection on traits, on climatic traits. And also we can consider the parameters being estimated. So instead of estimating, say, the minimum and the maximum separately and for each variable independently, we can try to find some parameters that are actually mathematically independent, yet represent a distribution of traits in multivariate trait space. So returning to the generalized least square approach, again here this last equation is we want to estimate the ancestor nodes here. And from this generalized least square, we have this error structure, epsilon, where we can incorporate the evolutionary model. And we've incorporated an evolutionary model of Brownian motion. And basically, Brownian motion is represented by the change in a trait by some variance of the trait, or the evolutionary rate, times some white noise. So this Brownian motion is also represented in other models that incorporate selection, such as an Ornstein-Uhlenbeck process. This model also has the Brownian motion as a term here, but it adds this extra term that includes a selection parameter, alpha, which is multiplied by some distance from an optimal trait, so an optimum in trait space, which is theta here. So we can incorporate this model into the error structure, and it's been done to get better models of ancestor states. And the other thing we can consider is the actual parameters that we're estimating. So I'm going to illustrate an example for you in trait space of how I think we might be able to obtain better parameters. And I would really love any discussion that you have on these parameters or any suggestions as well. So this illustrated example shows two species and two traits. So we have trait space here. This is trait X on the x-axis and trait Y on the y-axis. And the two species are represented by the colors, black, one species, and gray, another species. So you can see that these distributions are heteroscedastic. And you can see also that they have unequal covariance. In fact, their covariances are opposite. So looking at a traditional reconstruction of ancestor states, if we were to reconstruct the mean of each trait separately, represented the observations, or the extant observations are represented by the red dots here, and apply some sort of confidence interval to that, we might estimate the ancestor to be somewhere in between. So this is an ancestor estimation. And then we can apply some sort of confidence around that estimate. Now, of course, the confidence that you apply will depend on how much trait space that you want to occupy by the ancestor. But this estimate doesn't incorporate anything about the covariance between these two traits. The method that I've used, the max-min coding method, uses, of course, the minimum of each trait as well as the maximum. That's of trait X. And looking at the minimum of trait Y and the maximum of trait Y, we can chart in here some estimate of the trait space that the ancestor might occupy. So this is the climate envelope that the ancestor might occupy. OK, so the parameters that I think we might be able to use to estimate really the covariance structure between the ancestor covariance structure between these traits would be these parameters here. So the mean, which has already been illustrated, some standard deviation or some measure of variance of these distributions, as well as a rotation. So if we use singular value decomposition, we can estimate the major axis of each of these distributions. And then we can calculate the rotation between the major axes of these distributions. Now, trying to apply this method, I've made a discrete time implementation stepping through a translation, rotation, and scale in trait space of these parameters. So here's an animation of this distribution, translating, rotating, and scaling to another distribution, and then back to its original distribution. And if we stop this somewhere in between, we might get some estimate of where we think the ancestor distribution might occur, or the space, the distribution in trait space that the ancestor might occur in. So if we map on top of this, the more traditional estimate without covariance, as well as the climate envelope, we can see that in trait space, these estimates give these different parameters with the same method will give very different estimates in trait space, and that's due to taking into account covariance. So to move this forward, I'd like to extend this to an in-trait, in-species system. I'd like to make some evolutionary simulations using both a neutral model of evolution as well as a model that incorporates selection. And I'd also like to see how this can apply to other disciplines or other subdisciplines, such as geometric morphometrics, because shape has a lot of covariant structure built into it, or the analysis of landmark data points. So this can have some implication for the evolution of shape. OK, drawing you back to the first part of the talk, I discussed using past geographic responses to inform future expectations of responses. I used a paleophylogeographic species distribution model that is a geographically and temporally explicit model of suitable habitat, incorporating two modeling components, which is a changing climate as well as phylogenetic effects. And I showed implementations of this model at two timescales. At a short timescale, looking at glacial, interglacial fluctuations that showed very little phylogenetic effects, and also at a long timescale that probably incorporates more phylogenetic effects, but we are not able to validate these models because we don't have, I didn't implement the various models of evolution, as well as looking at different parameters that we could actually model through time. So I'd like to thank David Polly, Amelia Martins, Dennis Reuter, and Brian O'Meara for contributing discussions to various theoretical aspects of this work. And I'd also like to thank Amael, Chris, and Claudio for sitting through this presentation yesterday and giving me some excellent feedback. And I would like to acknowledge the NSF and NIMBioS for providing support for this work. So, thank you.
Yeah, so you asked the question about how to model the distribution in trait space and perhaps a better way of mapping one species onto another. And I think that the normal way to do this, or perhaps the most straightforward way to do it would be to, rather than to use min-max, boxes, cuboids, but to use, assuming a Gaussian, a multivariate Gaussian distribution, to model it simply as a multivariate Gaussian, or two multivariate Gaussians, and then you can use Bayes' formula to work out posterior probabilities, and then you don't have the problem of SVD in finding a major axis, minor axis, because the covariances are all built into that multivariate distribution. And you said you didn't want to discuss other models other than the bioclimb. But
well, I wasn't going to present any other models besides the bioclimb.
Okay, but you implied that bioclimb is the best or the most accepted.
It's the best when you're looking at non-analogous climates. But if you're only looking at modern climates, there are other models that outperform this.
Okay, well, I don't know what the other models are. But again, I'm surprised that people use this cuboid approach when certainly looking at the data, I don't know if that was cartoon data or real data, but certainly looking at the data, you're much better off with a multivariate Gaussian description of the data points. And I think that might help.
Right. That's what I'd like to get to, is a multivariate description of these distributions in trait space.
I mean, it's pretty easy to do, so talk about it tomorrow.
Okay, excellent.
On the last kind of animation you had with the rotation, is there some reason why that makes more sense than, say, the minor axis elongating and the major axis shortening, so they kind of switch roles as it deforms into the other one? Like, is there some reason to believe that somehow the longest axis will remain correspond to the other longest axis?
No, I think that you could also, Chris and I were discussing this yesterday, I think you could also use some measurement of the spread of each trait within a species. Is that what you're talking about? Actually changing a parameter, which would be the spread of the trait, as opposed to the rotation of
the I was picturing that thing deforming by just somehow the axes deforming in length as opposed to rotation.
So thinking about when these distributions deform, we would want to maintain, like, say, a Euclidean distance that's calculated between all the points within the distribution, therefore not to deform the climate space or the trait space.
So like a...
So we want a rigid rotation.
Yeah, right, okay.
Yeah, not a...
And is there good evidence for that makes sense? I mean, to me...
Well, whenever I'm thinking about applying this across disciplines, like, so in climate space, you might be able to deform the climate space without maintaining the distances between the points. But if you want to apply this, say, to a morphometric space, you should not deform the distances between the points within within the landmark data. So I guess we'd have to decide what is the trade-off, broad application or, right.
Just in looking at your projection of what was going to happen with the rattlesnake, I was kind of expecting as the climate warmed that rattlesnakes would be moving north.
Right.
And yet they didn't manage to do that.
Right.
Was there an explanation for maybe climate changing too fast or something along those lines?
Right. So in this particular projection, if you look at the 1 degree increase, the distributions do move north. But if you look at the 6 degree increase, you have a lot of contraction and fragmentation. And that's due to the multivariate nature. So I'm just reporting the 1 degree or the 6 degree increase on this slide. But the other variables in climate space aren't changing necessarily linearly with the temperature. So when we get such a hot climate, it doesn't mean that it's always good for these rattlesnakes, because other variables are responding differently. But it's not about the rate, really. I mean, it is about the rate, but if we make these the projections don't depend on a rate. They just depend on where can we find the suitable habitat of this species on this particular climate.
When you're talking about using the geographic center approach,
Yes.
and again, looking at the rattlesnakes, where you ended up, at one point, it seemed like there was a little population down towards Texas, and then
Yeah.
there was one up in the Appalachians.
Right.
How does the geographic center approach deal with having those disjunct populations?
Right. So I've also simultaneously calculated the area, the areal extent, which would not consider some combined area, just the actual points in geographic space. So it would be greatly influenced by both of these distributions for the geographic center. But if you consider several of these quantifications of geographic displacement, such as areal extent, that would be, in that case, for the timber rattlesnake, that would be a better quantification of how it changes through time.
Thank you.